I'm Dr. Pam Ventola. I'm a Senior Science Director at Cogstate, and I lead all of our rare disease and pediatrics work. I'm going to talk about a recent poster presentation that we presented at the NORD Summit on a Rater Academy approach to rater training. That's a highly efficient yet effective way of training raters across a program of clinical trials. So in rare disease trials, the sample sizes are often very small because there aren't often many patients with the condition, hence it's a rare disease. So every data point is highly critical for rare disease trials, and therefore making sure each data point is of high quality is of the utmost importance. That being said, there's also often highly specialized sites in rare disease trials, so these sites are participating in multiple trials. Therefore, as a field, it's very important for us to increase the efficiency in how we're training raters across clinical trials. So this presentation is some data that we collected on how to train raters both effectively but also highly efficiently through a Rater Academy program. So the instrument that we used for this project to illustrate the effectiveness of the Rater Academy approach was the Vineland Adaptive Behavior Scales. The Vineland is an interview done with the clinician and a caregiver, and it assesses the patient's functioning both at home and in the community. What's interesting about the Vineland is that it can be very complex to administer and score, yet the qualifications required to administer the scale are lower than some of the other neuropsychological assessments that we use. So the Vineland's often administered by a study coordinator or junior staff member, which makes the training component for the Vineland of particular importance. So what we did was we developed this highly efficient training model called Rater Academy for training clinicians on the Vineland. And it's completely remote. All of the training was done online, which also is highly efficient. So no one had to travel to an in-person meeting for training. It was all done completely remotely. So the clinicians that were participating in this Rater Academy program completed a comprehensive training program on the Vineland, which took about 90 minutes. It was completely web-based. There was an interactive module that covered clinical rapport, administration practices, and scoring. This module included interactive knowledge checks, like quizzes. There were video demonstrations and case examples. The clinicians, as a next step, then completed a Vineland 3 with a mock caregiver, so it was a practice Vineland. They audio recorded this interview and submitted the audio recording as well as the source document, which is the record form, for review. And then a neuropsychologist reviewed this audio recording and record form and provided written feedback to the rater about administration and scoring practices. If any errors were identified in this practice administration, the rater had a one-on-one training call with the reviewing neuropsychologist. After a successful mock administration, after the neuropsychologist said the administration was perfect, the rater was then certified for the trials. So 52 clinicians completed the Rater Academy training program. Of these clinicians, 48 of the 52 completed a practice administration accurately with no errors on their first submission. 4 clinicians required that one-on-one follow-up phone call with our neuropsychologist. And then following that phone call, all of them, all 52, were able to be certified. So all these 52 clinicians were certified in the program. Then 27 of the 52 participated in one trial, 20 participated in 2 trials, and 5 participated in at least 3 trials. So of these clinicians, out of these 52 clinicians, They administered 138 Vineland administrations across the program. So across these 4 trials, 138 Vinelands were administered. We also did a check of the in-study. So of these 138 administrations, we did a random check of the accuracy. So we reviewed 53 administrations of these 138 that were identified completely randomly. So 53 were selected for review. And of these 53, there were only 4 that had significant errors. Following these 4 administrations, there were 3 raters that did these 4 administrations. Each had a one-to-one phone call with a reviewing neuropsychologist. And for the rater who made the 2 errors, after the second administration, the neuropsychologist had a call with the principal investigator at the site. And then after each administration where an error was identified, we then reviewed the subsequent administration, and they were completely fine. So with the rater that had 2 errors and 2 administrations, we then reviewed the next 2, and they were also fine. So our remediation efforts led to, following that, led to perfectly administered Vinelands. So, overall, this model was highly, highly efficient. So we trained 52 clinicians in about, each spent about 90 minutes in the training process. And then they were then certified not just for one trial, which is the traditional model, but they were certified across the whole program. So this kind of a model, this Rater Academy model, greatly reduced training time. It was highly efficient in the sense that the raters were then certified immediately for 4 possible trials, which was all of the trials in the program. And it was effective. So of the administrations that were reviewed, the majority of them were perfect. And then the ones where errors were identified, we did a follow-up, and the subsequent administrations also were perfect. So this Rater Academy model definitely is one that we should use moving forward in rare disease trials specifically. And expanding the model will support the scalability of rare disease trials and allow the sites to have high data quality. The sponsors will receive high data quality, but will allow the sites to have more efficiency in their training process, which will reduce cost, reduce burden, and timelines for study startup.